Hey everyone, this video is going to cover the sickle cell lab that is put out by Advotech. It's one of our 3 basic gel electrophoresis labs that we're offering over this program. And I think that this is a great lab to actually start your class off with when learning the process of gel electrophoresis. So out of all the 3 gel electrophoresis labs that we're offering, the HNPCC lab, the cancer detection one, the sickle cell lab, and the DNA damage and repair, this one is probably the easiest to incorporate into your classroom. It's got the fewest number of samples, and also Advotech has made it very easy if this is the first time you're doing gel electrophoresis with your class to actually make sure that we load these samples properly into our gel. So the premise behind this lab is we are going to look at 3 individuals, a mother, a father, and a child, and try to determine who has the sickle cell trait based on their genotypes. And these samples are pre-digested, so there's nothing that you have to do to treat the samples, and the samples also have loading buffer with them. So all your kids have to do is really pipette the samples out of the container that they come in, and then load them right into the gel and then visualize their results. So this is really a good lab to start your class off with if they've never done gel electrophoresis before. So what you're going to get, a box that looks like this, and this is all for a full class, so it's very, very compact. And in here, you're going to get some even pipettes that you could load your gel with if you don't have the micro pipettes that I'm going to use in this video, and also that have been given to you in your Baxter box. But in case if you don't have those, there are disposable pipettes in here that you can load your samples with. Other things that you're going to get, the actual DNA samples that you would distribute to each lab group, so they're going to come in a strip like this, and this is something that's very unique to Advotech, especially if you're doing this with younger kids and they've never done this before. It kind of puts all their samples in order, so they will have a proper ordering when they put them in their gel, and they'll know exactly what order they put everything in. So this is really nice. So basically you would just cut off a strip per lab group and give them that to load in their gel. Other things that you're going to get, agarose, which I'm not going to use today just because I would have to probably weigh this out, and I don't have a balance at my house, but I'm going to show you something else that we can do that's really easy. Some stain for the end of our gel, and I'm actually going to run 2 gels for you today. One I am going to stain and show you how the stain works, but another way that I'm going to run the gel, we're going to be able to visualize our results immediately using this blue gel system, which is really, really nice. So I'll talk about that in a minute. The other thing that you're going to get is some gel electrophoresis buffer, and this particular buffer is at a 50x concentration, so I'm going to tell you how I'm going to dilute this, and just in general how you can dilute a lot of different concentrated buffers. Also, what is included in the kit, which I'm not going to use in this video, is some practice loading dye that you could always give to your kids in class if you wanted them to just practice loading gels before you actually run your real gel for the sickle cell lab. And then also some InstaStain blue strips that would be used to dye your gel and visualize your DNA at the very end. And I'm not going to use these either, but we can talk about this when we have our office hours if you have questions and want to try this out. So when we're doing this lab, there's 3 main things that you have to do. The first one is to make your buffer. The next one is to make your gels, and then the third is just to load your samples and actually run your gel. So I'm going to make a buffer with you for right now, and whenever you're making a buffer, you always want to use the equation C1V1 equals C2V2. And if you do that, you can pretty much dilute any buffer that's given to you. So the buffer that you may have received in your Baxter box was a 5x concentration. That means it's 5 times as concentrated as it needs to be. The one that came in the sickle cell lab is 50x, so it's 50 times as concentrated as it needs to be. So it doesn't matter which one that you use, but I'm going to make enough buffer to actually run my gel and then also to make my gel. So this buffer system, or gel electrophoresis system that we're going to use, is the blue gel system that is put out by a company called Mini PCR. And it's really awesome because what it can do is we can visualize our results immediately. And if you're in the classroom and you've ever run gels before, you know that the visualization a lot of the time can definitely be a bottleneck in your classroom. So this whole system is going to get rid of that for us. So just to run through the parts of this is going to be our gel casting tray, which is really, really nice. This white plastic here is heat resistant, so it won't warp even after we make our agarose gels. So once you've melted our agarose, which I'll do with you in a minute, you can pour your gels immediately. Then we have this, once you make your gels, you're going to take out this little gel tray and you are going to put it in this buffer container, which fits in nice and easy. And then this buffer container is going to go in our electrophoresis box. You can see there's electrodes at the top and the bottom here. And this cap is just going to hook on top. And once we plug it in, we'll provide that current that's going to allow our DNA to run from the negative side to the positive side. And with this system, there is a light incorporated into it, and I'll show you that in a minute once we get our gel running. But this will allow a blue light to come on, and then it's going to basically fluoresce our DNA bands. And all we have to do is put this little hood over it. It's kind of like a dark room. Which I will show you once I start taking pictures of our gel. And then we will be able to visualize our DNA bands immediately. The kids will just pop this over and then they can take a picture with their cell phone. So this is really, really, really convenient. Okay, because of that, the reason why I'm introducing this now is because how big your system is really depends on how much buffer you want to make. So if you do have gel electrophoresis equipment at your school, but it's not this blue gel system, it's probably a lot bigger chamber and you're going to have to make more buffer than what I'm making right now. But I do know that to pour 2 gels, which I'm going to do for this, they each need about 20 milliliters of agarose for our gel that we're going to make. And then this buffer system, or this buffer chamber, it probably holds about 30 milliliters or so. So I'm going to run 2 gels for us. I'm going to make 250 milliliters of buffer, and that's going to be plenty of buffer in case if I mess something up, but it will also be enough for me to make 2 gels and also to run both of those gels. What I am going to use for this lab, if you did watch the HNPCC video, I used the 5X buffer that the Baxter ladies gave to me, but today I'm going to use the 50X gel electrophoresis buffer. And if I use that equation, C1V1 equals C2V2, if I use this 50X buffer and I want to make 250 milliliters of buffer, my C1 in that concentration, or in that equation, my C1 is 50X. My V1, that's my unknown. I don't know how much of this I want to use. And then I want to make 250 milliliters, that's my V2, with a 1X concentration of this. So basically, I've got a 50X concentration that I'm going to dilute down to a 1X in 250 milliliters. That means I'm going to use 5 milliliters of this and 245 milliliters of water. So I'm going to do that now. And one thing that I'm going to use is the spring water that I purchased. So you do not want to use tap water. That's the big thing here. So I'm just going to measure out 245 milliliters. And it doesn't really need to be super exact, but we'll see how close we can get it here. So I've got 100 there. We'll do another 100 here. And then another 45. Okay, pretty close. All right, for the 5 milliliters of the concentrate buffer, instead of really just trying to pour that out, a lot of times you can use a transfer pipette that has graduations on it. So the one that came in the Advotech kit, if you really look closely, you can see up the stem is 3 milliliters. There's also probably some other graduated pipettes that you got with the BioRad kit we used for the algae beads. That stem is a lot thinner, so it's only 1 milliliter. But all I'm going to take now is my 50X gel electrophoresis buffer, and I'm going to take out 5 milliliters. here. Actually, this might be easier to pour in here. Didn't come up as nice as I wanted it. But what I can do with this, if I measure out 5 milliliters into my graduated cylinder, I can always use this in case I'm not very exact. So in this case, I now have my 1X buffer solution. So again, that was 245 milliliters of water and then 5 milliliters of my 50X concentrate buffer. If you want to use the 5X that was put in your Baxter box, you're going to use 200 milliliters of water and 50 milliliters of your 5X concentrated buffer. So here's my buffer, and now I'm going to get ready to make my agarose gels, which is step 2. I want to make 2 gels, and I know that each one of these holds about 20 milliliters of agarose, so I'm going to make 50 milliliters of agarose just to make sure that I can pour my gels pretty deep. So that being said, I'm just going to use my graduated cylinder again. I just had water in here, so it wasn't concentrated buffer. And after I give this a good swirl, I'll measure out 50 milliliters. Probably should turn it towards me so I can see it, right? Okay, 50 milliliters. And I'm going to pour it into this little 100 milliliter flask here. Now, in the Advotech manual, they recommended a 0.8% agarose gel. I'm actually going to make a 1% agarose gel. The only thing that changing the percentage does is the higher the percentage of agarose, the smaller the pores. And that means if you're running a gel that has small DNA bands, you're going to get better separation if your percentage of agarose is higher. And in all honesty, it's not really going to make a difference between a 0.8% and a 1% gel. So once we get into our PCR labs, you might see that we're running higher percentage gels, like a 2% gel, and that's just because our DNA bands are much smaller. So we need to get better separation by increasing that concentration of agarose and making our pores smaller. In this case, the reason why I'm making a 1% gel is because I have these nice little tabs here that are already measured out at 0.5 grams. And 0.5 grams in 50 milliliters of buffer is going to give me a 1% gel. If I wanted to make a 0.8% gel, I need to use 0.4 grams. And like I said, I don't have a scale at my house. And this says 3 grams in here, and this is 0.5 grams. So the chance that I'm going to get 0.4 grams is going to be highly unlikely. So I'm just going to make a 1% gel. I just might have to run my gel a little bit longer, but I'll show you how we can check that. So I'm just going to pop this open. And this agarose tab is really, really handy because, and yes, I should be wearing gloves, but in all honesty, the gloves that I have are too big and I tend to drop things. So I washed my hands before and I'll wash my hands very well afterwards. So with these agarose tabs, you just want this to dissolve for about 4 minutes. And once it dissolves, we're gonna pop it in the microwave and we'll melt it down till this is a very, very clear and transparent solution. So right now you can see it's kind of turning opaque. I'm gonna let this dissolve for about 4 minutes and then I will pop it in the microwave. Okay, welcome back. So I just got done melting my agarose. I threw this flask into the microwave for 45 seconds and you can see that now my agarose is extremely transparent. If you're a little bit worried about throwing this in the microwave for that long, you can always try a lesser time, like 30 seconds. As soon as this looks very transparent, then you are good to go and you don't need to microwave it any longer. I'm kind of a wuss, so I'm definitely handling this with hot pads. These have kind of become my designated lab hot pads around my house, but I definitely recommend that you do so. And if you touch the flask a little bit, you'll see it's pretty hot. But like I said, I'm kind of a wuss, so maybe that's not too hot for you. I'm going to pour both of my gels now, and for this particular lab, you only do need to run one gel unless if there's something that you want to practice. So the reason why I'm running 2 gels, I'm going to get the exact same results, but one gel I want to stain with this concentrated stain because many of us might not be able to visualize our results immediately once we're back in the classroom if we don't have this blue gel system, or if we don't have a blue light or UV transilluminator, and I'm going to explain that in a minute. So the results and everything about these gels are going to be exactly the same. It's just one I'm going to be able to visualize the results right away because I'm going to use a special dye that I'm going to put into the agarose, and then the other one I'm going to have to stain the gel after we run our samples, which is going to take a little bit more time. So that being said, I'm going to pop the combs for these gels are on the bottom of the tray. So I'm going to take both combs out because I'm going to be running 2 separate gels here. And you will see that the combs that are offered to us, we have one with a ton of samples. We're going to be able to use the combs that have lesser samples. We have 6 samples in this lab. We've got 3 controls, and then we have a mom, a child, and a father that we're going to test. When we look at this casting tray system, one thing that's really nice about this is the actual gel tray has a different number of ridges on either side. So on this side there's 2 ridges, if you can see here, and on this side there is 1. What is nice about that is it only fits in the gel casting tray in one particular order. So when I do that, what can happen is I'm going to place my combs into the gel casting tray at the top. So no matter what, when I take this gel out of this casting tray and I put it in the gel electrophoresis chamber, or the buffer chamber, what's going to happen is this tray that I have my gel in is only going to fit in this buffer chamber in one particular orientation. And therefore, it's always going to make my wells holding my DNA samples towards the top. And when we put this buffer tray into our gel electrophoresis system, again, my samples will automatically be on the top, so the kids can't mess up the orientation. For example, putting the DNA bands down low where our positive electrode is going to be, because you know if that happens, then they're just going to run their DNA samples off the gel. So one thing that I do really like about this whole system, especially for first-time users, is they don't really have to worry about messing up the orientation of their gel in relationship to the electrodes. So I'm going to place my combs into my tray, and I'm going to pour my first gel. Now my first gel is just going to have this agarose that I melted in it. So I'll go ahead and I'll pour. Again, this white tray is heat resistant, so we don't have to worry about any warping. And I'm going to pour about halfway up the combs. Okay. One thing too is that this gel tray is not going to leak. This particular gel is going to be the one that I'm going to stain with this blue dye at the very end. However, there's this other thing that you should have gotten in your Baxter box, and it is called a gel green stain. And this stain is going to stain our DNA bands, and with the blue light that this gel electrophoresis system allows, we'll be able to visualize our bands immediately. Now this gel green stain is at a 10,000x concentration. So right now, I'm not exactly sure, but I probably have anywhere from about 20 to 30 milliliters of agarose in this flask. So what I'm going to do is take 3 microliters. I'm just going to err on the side of caution and say that I want to make this more concentrated than less. And I'm going to add 3 microliters, of this stain to my agarose. So I've already got my pipette set to 3 microliters, and this gel green stain here is light sensitive. That's why we have it in that little black baggie. And I'm just going to take out a very small 3 microliters, so I don't even know if you can see that. And I'm going to add it directly to the agarose, and it's going to turn a little bit of an orangish color. See, I had to waste a little waste cup over there. So again, this gel green stain is 10,000x concentrated. So 3 microliters into 30 milliliters is going to get it diluted down to about where it needs to be. And I'm just going to give it a little swirl, kind of get it evenly distributed. And by now my agarose is cooling a little bit. And so I'm going to pour my second gel with this gel green stain. I'll pour it about halfway up the combs, maybe a little bit deeper if I can. All right, perfect. Okay, and now I'm just going to let these 2 gels sit. I'll check them maybe in about 15-20 minutes, and then we will load our samples and run our first gel. Okay, and we're back. So I'm going to load my gel now, and the gel I'm going to run first is the gel that we had stained with the gel green dye, so we can visualize our samples immediately. So you can tell between these 2 gels in the casting tray, the one down below is the one that had the gel green dye in it. You can see it's definitely more of a yellowish tint, and I can just tap my gels too to make sure that they're solidified and they're good to go. Just be careful not to poke them. And what I'm first going to do is gently remove the comb. So I'm just going to wiggle it a little bit and kind of get a feel for it, pop it out right there, and you can see that we had really, really nice well formation. So I'm happy with that. I'm going to take this gel out of the casting tray, and you will see that it will fit very nicely into our buffer tray of our blue gel system. So there's 2 ridges on this side. You're also going to have 2 ridges on that buffer tray, and it will fit in very, very nicely. Now, even when I run my second gel, the one that I'm going to stain afterwards, all of this is the same. So we're not doing anything different between these 2 gels. So I'm not going to repeat this process for you. But again, I have my buffer that I made previously, and I'm just going to nicely pour that over my gel. Oops, that was a little high, but we should be okay. And put that off to the side. And then I'm just going to start loading my samples. So again, I said that one really nice thing about Advotech is their samples come in strips that are already in the proper order that you want your kids to load the gels. So in class, every lab group would get one of these strips. And all the kids have to do is take a pipette tip. So Advotech in their manual, they say 35 microliters of sample into each well. My pipette only goes up to 20. So I'm going to set it at 20 now, and then I will see if I can still fit more into my well. So I tried to pour these wells as deep as I could. So you just pretty much poke through the actual like aluminum foil covering, and you get your pipette all the way down into the sample. So I think I don't have the best visual there. Let me turn this around. So if you can see, my pipette tip is all the way in the sample. So I'm just going to press down on my pipette, and I'm going to soak up as much of the sample as I can. I might pipette up and down just a little bit to mix the sample. And let's just load my first well here. So your first 3 wells are going to be control samples, one being what a sickle cell gene would look like if you're a homozygous individual, one would be if you're a carrier, and one would be if you don't have sickle cell. And then we have, like I said before, a mother, a child, and a father. Now I still have some sample left in this strip, so I might just try add what's left over, but I highly doubt I need more. And let's see. One nice thing about this Advotech kit that is a problem with the Carolina kit is there's plenty of sample, and it's all down on the bottom of the little wells here. The Carolina kit, it was a problem getting some of the samples out. So I almost have another full 20 microliters here. I don't think I'm going to be able to fit that into my well, but I'll try get a little bit more and see if I can pack it in there really tight. Okay, so looks pretty good. Okay, so I'm going to load the rest of my samples here. Okay, sample B is going to go in well 2, sample 3 in well 3, D and 4, E and 5, and F and 6. I'm going to change my pipette with each sample, and then all I'm going to do is pop on this cap and I'll turn it on. So I'll tune back in with you in 1 second. Okay, so I have all my samples loaded here. So all I'm going to do is pop on this cap, and these electrodes in the cap are going to match up with the electrodes in the gel electrophoresis chamber. You should have a cord to plug in your gel blue system in the back. So the plug-in is right here. For this particular system, we are not setting a voltage, and we're probably going to run our gel for, I don't know, maybe 20-30 minutes. But the awesome thing about this is we can keep checking in with it to see how far our bands have run. So this is going to be our power button. I'm just going to hit this here. Once you see that green light go on, then you can check off to the side to make sure that you have some bubbles coming up in the buffer chamber on the negative side. And I will just keep checking in with this to see how far my bands have progressed. How I'm going to do that, this button right here allows a blue light to shine up through our samples. And when we put on this dark room, that little cover that I was talking about previously, all we have to do is pop this on over the top. So it might take me a second here to fit it on because we do want it to fit over, there we go, the orange part. And then the kids can just put their cell phone right there and they can view their samples. So I will take some pictures for you about every 5 minutes or so, or when I've actually seen some progress. The second gel that I'm going to run, because there is no gel green in the gel, this whole part is going to be pointless. So we aren't going to be able to really see our bands as much with this blue light. So what I'm going to do for this next gel, so I'm going to continue to let this run, but I'm going to shut this light off. I'm going to do the exact same thing that I did with this gel green gel. I'm going to take this gel out, I'm going to put it into our chamber, I'm going to load our samples, but then I'm going to take the gel out and I'm going to stain it. So I will check back in with you once this gel is done running and I'm ready to stain my next gel after I've loaded my samples and run that gel. Okay, so for the last part of this video, I just want to show you how we can also visualize our DNA results by staining our gel. So currently I have my second gel running. I added the buffer, loaded the samples just as I did with the other gel, but this gel did not have that gel green stain added to it, so I can't visualize any results by turning on this blue light and looking through that little dark room hood that I had put over this gel electrophoresis chamber previously. So what I'm going to do, first I'm going to make my stain that I will use in a moment to stain this gel. And to do that, for this part, I am going to put on gloves, just because this stain can actually kind of get on your hands and take a little while to come off. Now, if you are going to do this, please just be careful using this stain. You definitely don't want to get it on any of your household furniture or your carpet or anything like that. So if you don't want to do this part, it's totally fine, but at least you'll have this video for later reference on how to do that. So this stain is a 10x concentrate. So using, again, my equation C1V1 equals C2V2, this is a 10x concentrate, and I want to turn it into a 1x concentrate, and I'm going to make 100 milliliters. So to do that, I'm going to take 10 milliliters of this concentrated dye and add it to 90 milliliters of water. So I'm going to use my spring water again, and I'll measure out 90 milliliters here. I'm going to use a little bit of a bigger flask than I did to make my agarose, even though I'm not using that much volume still, just because I definitely don't want this to spill. So I'm going to use a 250 milliliter flask. It doesn't matter, you can use something smaller. Like I said before, I'm just kind of clumsy, so I want to make sure I have a big enough flask here. So I have 90 milliliters of water, and now I'm just going to add 10 milliliters of this stain. I'm not going to use the graduated cylinder because I don't want to get that really crazy with the stain. And to be honest, this is looking pretty dark already, and I've only used 4 milliliters. So I actually might stop at around 8 milliliters here. So this pipette that I'm using does have graduations on it, and I've gone up to the 2 milliliter graduation mark 4 times, and I'm just going to leave it at that. Usually if you make a weaker stain, it's totally fine. It will still work. You just need to stain your gel for longer. So with this concentrated, it's just a little bit less than a 1x. I'm going to, when this gel is done running, just slide it out of the chamber after I turn the power off, and I will put the gel in this staining tray that I have here that should have come with some of your kits, or hopefully they put in your Baxter box. And then I'm just going to make sure it's completely submerged in the stain for about 5 to 10 minutes. I'm going to put some spring water over it to de-stain it for about 20 to 30 minutes, and it will be something you just have to keep an eye on. And once I've done that, I'll probably be able to get a good picture just putting it against a white background. For example, if it's in the staining tray. Otherwise, if you're at school, you just kind of need a white light to illuminate it. So again, I'm going to pour this stain that I just made over my gel for about 5 to 10 minutes. I will pour that off into something that, you know, I'll make sure is not very porous and it won't stain. And then I'm going to pour spring water over it to de-stain it for about 30 minutes and take a picture for you. And I will post that. All right, guys, I hope that was helpful, and I will see you later this week.